Now starring on Biology for Brainiacs, we have two very special guests whom you've heard before, Matthew Meselson and Franklin Stahl.
Thank you so much for having us.
The pleasure is all mine. Let's get to why you're here. We all know about your amazing discovery in regards to DNA, but tell us about how you first got into the field of biology. I was born in Boston, Massachusetts, where I ended up getting my BA at Harvard University in 1951. And while finishing up my PhD, I attended a molecular biology course at the University of Rochester, which was taught by the famous duo James Watson and Francis Crick.
This is also where Stahl introduced me to Watson and Crick. At the time, I was a graduate student at the California Institute of Technology, where I was interested in exploring new methods of experimentation. With Stahl's experience and skills in mathematics, he helped me to design these experiments. We hit it off right away.
Now that we know about your history, let's talk about the well-known meselson-Stahl experiment on semi-conservative DNA. What were your steps leading up to this amazing discovery? After Watson and Crick discovered the double helix structure of DNA in their paper published in 1953, we had so many questions, but we were most concerned with the replication process of DNA. The three models we were testing were the semi-conservative replication, conservative replication, and dispersive replication models. In the semi-conservative replication model, the two strands of DNA unwind from each other, and each acts as a template for synthesis of a new complementary strand. This results in two DNA molecules with one original strand and one new strand.
In the conservative replication model, DNA replication results in one molecule that consists of both original DNA strands identical to the original DNA molecule, and another molecule that consists of two new strands with exactly the same sequences as the original molecule.
And in the dispersive replication model, DNA replication results in two DNA molecules that are mixtures or hybrids of parental and daughter DNA. In the model, each individual strand is a patchwork of original and new DNA.
We conducted our experiment on DNA replication using E. coli bacteria as a model system. We began by growing E. coli in a medium or nutrient broth containing a heavy isotope of nitrogen. When the E. coli was grown on this medium, the bacteria took up the nitrogen and used it to synthesize new biological molecules, including DNA.
After many generations of E. coli growing in the heavy isotope of nitrogen, the nitrogenous bases of the bacteria's DNA were all labeled with heavy nitrogen. Then the bacteria were switched to the medium containing a light nitrogen isotope and were allowed to grow for several generations. The DNA made after the switch would have been made up of light nitrogen, as this would have been the only nitrogen available for DNA synthesis.
We knew how often E. coli cells divide, so we were able to collect small samples in each generation and extract and purify the DNA. We then measured the density of the DNA using density gradient centrifugation.
For our viewers, do you care to explain the centrifugation method?
Yes, of course. This method separates molecules such as DNA into bands by spinning them at extremely high speeds in the presence of another molecule that forms a density gradient from the top to the bottom of the spinning tube. Density gradient centrifugation allows for very small differences, like those between heavy nitrogen and light nitrogen labeled DNA, to be detected.
So now that we have an understanding of the experiment, let's talk about what the results proved to you guys through the first few generations. The DNA isolated from cells at the start of the experiment, known as Generation 0, just before the switch to the light nitrogen medium, produced a single band after centrifugation. This made sense because the DNA should have contained only heavy nitrogen at the time.
The DNA isolated after one generation, which was one round of DNA replication, also produced a single band when centrifuged. However, this band was higher, intermediate in density between the heavy and light nitrogen DNA. The intermediate band told us that the DNA molecules made in the first round of replication was a hybrid of the light and heavy DNA.
Information from the second generation let us determine which of the remaining models was actually correct. When we centrifuged the second generation DNA, it produced two bands. One was in the same position as the intermediate band from the first generation, while the second was higher. The results told us that the DNA was being replicated semi-conservatively.
And over the third and fourth generations, we'd expect the hybrid band to become progressively fainter, and the light band, which represents a smaller fraction of the total DNA, to become progressively stronger, which represents a larger fraction.
Wow, we thank you both so much for being on our show and your incredible contributions to the field of biology. Stay tuned next week for another amazing guest, Rosalind Franklin!